Okay, now we are going to move on to our responses to stimuli in flowering plants. And this is my favorite lecture because we go over my favorite topics. I love particularly root gravitropism, so we're going to get to that today. I will try not to be too long-winded. For this lecture, you are going to need your plant hormones chart, so make sure you go ahead and print that out so you can fill that in. So in terms of stimuli for plants, we're talking about stimuli that can be physical or biological. And we will call that the physical stimuli abiotic, so something not related to anything living, or a biological stimuli, which would be biotic. And for our abiotic stimuli, that can be internal or it can be environmental. And same thing with our biotic stimuli. They can be internal or environmental. So the responses to a stimuli are usually a growth difference. So if you just have a growth response, that's an increase in plant mass just by increasing the rate of cell division. But if it is growth towards or away from a specific stimuli, we call that a tropism. So you need to be able to list about 4 or 5 types of physical and biological environmental stimuli. So particularly on the left side, these are physical stimuli. So internally speaking, that would be like an electrical impulse. Environmental would be things like light for our phototropism. The amount of carbon dioxide in the air, remember we said that that creates a differential growth response in the fact that we either have more or less guard cells being produced. Plants also have to deal with temperature, with touch, with the amount of water that's present. All of these things are stimuli, physical stimuli for plants. Biological stimuli is more like, internally speaking, that's their hormones or a change in which genes are being turned on. Environmentally, it might be herbivores or pathogens or even microorganisms in the soil. So how do plants then actually respond to stimuli? We have, it's a 3-step process. The first is the perception. They actually have to be able to perceive the stimuli in order to respond to it. So stimulus is the receiving end of things, usually on a particular receptor. Then transduction. One protein talks to another. This is like for me, transduction is like the chain reaction that happens after the perception and before the third portion, which is the response. So the response is a gene expression that creates a new protein or allows for that tropism growth to occur. So perception, transduction, and response. Those are the 3 different steps for how plants respond to stimuli. So let's take a look at an example of a plant response. So let's start with an herbivore is causing damage to a particular plant. So this lovely caterpillar over here on the left, there we are. There is our biotic stimuli. Okay, so once that happens, the herbivore damage causes a release of what we call volatile compounds. And those compounds are going to do 2 different things. First, they're going to cause a reaction in the plant that the herbivore is on. Okay, so as soon as that hormone is released, those volatile compounds, they're types of hormones, they are perceived among the plant. So that is the perception. And then once those volatile compounds bind to that receptor, there's going to be a bunch of different transduction signals. So hormones are going to be spread to the rest of the plant to let it know that it's been damaged. In this particular example, there's a secondary response also, and that is that these volatile compounds not only affect the original plant, but are spread into the air, and they affect all the neighboring plants of the same species in particular. And so that volatile compound binding to a receptor is the perception. And in both the original plant and into the neighboring plants, that primary binding of that receptor then allows for gene expression to be turned on, new proteins to be made, and then, so that's our transduction. And then the response is that jasmonic acid travels in the phloem of all the undamaged tissue to induce these defense responses throughout the original plant as well as the yet unaffected plants. So the response is jasmonic acid production throughout the plant. Okay, so how do animals communicate internally? Well, we have two different ways. First is the nervous system, right? We send electrical impulses from one part on our body to other parts to let it know what's going on. And the second is our endocrine system, or our hormones. Well, plants also use hormones for systemic communication. By systemic, that means throughout the body or throughout the organism. So what exactly is a hormone? A hormone is a small organic molecule that fits into a receptor and causes a change. And by definition, a hormone is made and stored in one location but acts in a different location of the body or of the organism. So plants have all kinds of different hormones, and we're going to be looking at these today. So you're going to have them on your chart. You don't have to copy this down right now because we're going to go through each one, but just giving you an idea of what plant hormones look like, what they're called, what their uses are, that sort of thing. So on this particular chart, you can see that we have gibberellic acid, we have auxins, ethylene, cytokinins, and abscisic acid, or what we call ABA. Those are the ones that we are going to be looking at today. So let's start with auxin. Auxin is generally produced in all the areas on this image that are in red. And what you can't probably tell is that most of these are at an apices, and that's for a very particular reason. So auxin is involved in fruit ripening. So when you don't have auxin, you tend to not have ripened fruit. This is the response that auxin is most known for, or, well, I guess one of the things that it's most known for, and that is what we call apical dominance. So remember I said earlier that auxin tends to be produced in apices? Well, so in this image, the apical bud at the top produces auxin, and what that means is that it works over here on the lateral buds or the lateral meristems to prevent branching from occurring. So it allows the apical bud to be dominant. When we take away the auxin, or we chop off that apical bud, then the lateral buds and lateral meristems begin to produce lateral branches. So the apical dominance is removed. So that is something that auxin is very well known for, is for being involved in apical dominance. But here's something else that auxin is involved in that will become a little more important later as we go into tropisms, and that is cell elongation in the shoot tissues. So if we look at our image here, auxin will stimulate a proton pump, and when it does so, the protons are excreted to the cell wall, and a high level of protons then causes what we call expansins, which are other proteins, to become active and loosen the ties that bind the cellulose together in a cell wall, loosens that up so it allows for cell elongation, or it allows for that cell wall to stretch so that when more water comes into the cell, those cell walls stretch and we have a longer cell than we started out with over here. So that is how auxin is involved in cell elongation, only within shoot tissues though. So this is just an interesting side note for you. I don't know if you remember anything about Agent Orange. Agent Orange was used in the Vietnam War, and it was sprayed across the jungle to defoliate all those jungle trees so that we could see where the enemy was. It is an auxin mimicking hormone that causes the plants to grow so fast that they lose their leaves. Okay, so there's a helicopter during the Vietnam War spraying that across the jungle, and so just something interesting to know about that is that it is also a human carcinogen. So it effectively killed about 400,000 Vietnamese and produced about 500,000 birth defects. And it also affected our U.S. soldiers as well. Many that went to war in Vietnam have come back with lots of different cancers caused by Agent Orange. And here's just another interesting side note. Agent Orange was made in RTP. The company that created it was here, so many researchers and those who worked in those labs have also been found to have cancer linked to that as well. Okay, so let's move on to happier notes. Our second hormone is gibberellic acid, which is also called gibberellins, and they function to also elongate the stems. They cause seeds to break their dormancy and begin to grow, and they can also cause flower and fruit development. So commercially, they're used to make seedless grapes very large because they enlarge the fruit. So all these hormones pretty much are used commercially to improve our food supply. So here is just an example of gibberellic acid being used on these lovely plants on the left. You can tell the first two were not treated with gibberellins, whereas the rest were. And so they are of major interest to agricultural scientists, or what we call agronomy. And they do come, like I said, in a commercially available form that allows you to let your plants grow tall. And here's just some more pictures to show you that, looking at the different plants that were treated with and without gibberellic acid. And then, like I said, gibberellic acid in seeds causes fruit to grow larger and stems between the fruit to become elongated. So here's our lovely grapes. And here's some more. So if you look at the grapes on the right, you can tell that the fruit itself is much larger than the fruit on the left. So that gibberellic acid allows that fruit to grow larger and the stems between the fruit to become longer so that there's more space for them to grow. So next time you eat grapes from the grocery store, you'll know why they're big. Okay, moving on to cytokinins. Cytokinins promote cell division, and they also promote early embryonic growth. They work in conjunction with auxins to allow for differentiation of different types of tissues. So, for instance, if you have just a mass of plant tissue, which I have done before, and then you look at the auxin to cytokinin ratio, and depending on that ratio, you can take that undifferentiated plant cell mass and cause it to grow either roots or stems, depending on the ratio of auxin to cytokinin. So that's how plant tissue culture is controlled. Cytokinins also keep our plants young. So high cytokinin ratio keeps the plants staying young. So if we have genetically modified lettuce that produces cytokinins for a longer time, it tends to not wilt. So that helps us to keep our vegetables longer. If you've ever picked lettuce out of a garden and it was not genetically modified, then you know that it goes bad within a day. So that allows us to keep our vegetables fresher longer. Those without cytokinin or with low cytokinin, the plants get old. So it's part of the abscission process that we've talked about before. The decrease in cytokinins then continues to allow those leaves to get older. Okay, abscisic acid. I do realize I'm kind of going through these hormones a little bit quickly, but wanted to do that so we can get to the fun, important stuff of the tropisms. These are good too, and you have to know how they work in order to understand the tropisms, but that's just letting you know why I'm kind of pacing myself this way. Okay, so ABA. It is what's called the protection hormone. So the first thing it does is it keeps buds and seeds from breaking dormancy too early. So this is a mutation, and we call this the foolish seedling because in this corn, it doesn't produce abscisic acid. So you can see that the corn is already starting to germinate while it's still on the cob, so it hasn't even hit the soil yet. So the foolish seedling does not produce ABA, so therefore it doesn't have that protection, so it breaks dormancy way too early. And the second function of ABA is to close the stomata when the plant is in water stress. Okay, so this is something you've seen before, but I just wanted to go over it in terms of how ABA actually acts so that we understand the opening and closing of stomata in that light. So the perception then is drought causing ABA to bind to receptors in the guard cells. And so, let me get my little marker here. Okay, so here it is. ABA binds to the receptor, and then once it does, calcium comes in and causes potassium to leave. So that's the signal transduction pathway. And the response is that the water follows the potassium ions out of the guard cells and the stomata closes. Okay, so abscisic acid then acts as a controlling mechanism for closing the stomata. Okay, so ethylene is one of those hormones that is in the gas form. Kind of like we talked about earlier with the example of the caterpillar eating the plant and it had volatile hormones. Ethylene would be one of those. So its function is really to promote abscission, promote fruit ripening, fruit drop, and leaf drop. So in our example down here on the bottom, the holly plant on the left doesn't have any ethylene under this particular beaker, and so the holly leaves and the fruit are perfectly happy. As a fruit ripens, it emits ethylene gas. So if we take this holly plant and under the same beaker, put this ripe apple as it's ripening and producing ethylene, you note that all of the leaves have then fallen because ethylene promotes leaf drop. So this is just to show you with a particular strawberry the days after it's formed and then it's off to maturity. As we go from left to right over time, we have very small amounts of ethylene gas in the beginning, and then there seems to be a cyclical nature to the ethylene production in day 26 to day 30, with a slight trailing off, and then between day 34 and then especially at day 36, there is a spike in ethylene. And that spike is when you have the reddest strawberries, and then shortly after that they are going to rot. So this is what it means in terms of the fruit that we eat, not just the strawberries, but we're going to take tomato as our example. So tomatoes are picked when they're green. They're easier to transport with minimal damage because you can imagine red ripe tomatoes all in a box being on a truck. By the time you got to the grocery store, half of them would be rotted, and that would be a waste of fruit and a whole lot of loss of money to the farmers. So they are picked when they're green, and then once they arrive at their location, they are actually sprayed with ethylene gas and allowed to ripen in place. And then we have our ripe red tomatoes for commercial sale after that. So that's actually how a natural hormone from plants is used for our benefit. Whew, okay, let's talk about tropisms. So tropism, as I mentioned before, is a differential growth in response to an external stimuli. So differential growth being a faster rate of growth on one side and a slower rate of growth on the other side. And it can either be a negative tropism or a positive tropism. A negative tropism would be growth away from the stimuli. So for instance, this image over here on the right, that is a tree that's growing towards the sun. So its phototropism would be a positive tropism because it's growing towards the stimuli. But if we think of it in terms of gravity, this particular tree would have a negative tropism towards gravity. Does that make sense? Our roots over here on the left have positive gravitropism. So, excuse me, a positive growth towards gravity. Okay, let's start with phototropism. And we're going to look at phototropism in the shoots or the stems in particular. And that just means that there's differentiated growth results in the stem that causes the stem to grow toward a light source. So in this picture, that yellow arrow represents the direction that the light is coming from, and you can see the stems of this plant are all growing towards that light source. Okay, so how does the plant actually detect the light coming in from one direction? Well, that is a phenomenal process. To begin with, the plant cell membranes contain photoreceptors, right? That shouldn't be any surprise. A lot of times they have photoreceptors because they need to know where the light is so that they can start producing sugars, right? Okay, well, the photoreceptor protein contains a pigment that's called phototropin. And that responds to certain wavelengths of light, and particularly blue light. So during phototropism, for instance, that requires blue light, or light between 400 and 500 nanometers. And if you'll remember, we talked about when blue light was present, the stomata open so that the plant can then photosynthesize. So these same proteins, the same phototropin and photoreceptors are used in the process for opening the stomata as they are for just detecting the light so that we can have a phototropic response. So if you're looking at our image over here, you will see right here is our photoreceptor containing phototropin. So this is actually where the perception portion of the phototropic response begins. So looking at it in more detail, at the very beginning, the blue light comes in and hits the phototropin portion of the photoreceptor. And that causes a change in shape of that photoreceptor, which allows for a transfer of a phosphate group from ATP to the photoreceptor protein. So in other words, right here, where's my pointer? There it is. So the ATP comes in and ADP is going to go out. Oh, I cannot write with this thing. Oh, wow, really can't. Well, you can see that right here, ADP. And then there's a series of signaling events that follow this initial perception of light, but this is the perception portion of our phototropic response. Okay, so here we go. Here's the perception portion. Here is our phototropin and our ATP. We lose it turns into ADP because we've transferred a phosphate group. Just trying to get you oriented as to what you're looking at here. Here's our light coming in from one side. It's a particularly blue light because that's what phototropin responds to. Now, the transduction portion of this whole event has a lot of steps to it. But what I'm going to tell you is the overall story, basically. So what happens is that auxin within the plants begins to move. If you see that right here, it moves away from the side that has light, and it sets up an auxin gradient where we have lots of auxin on this side and very little on this side. So the portion of the plant that is away from the light has much more auxin. So if you'll remember, our auxin caused a loosening of the cell walls, which allowed for elongation of the cells, and that's the response. So we have cell elongation on this side, but not on the side the closest to the sun, and the resultant growth is that the stem then bends towards the light. Okay, so let's just look at this again from a different perspective. We have the perception, which is when the light stimulates the phototropism on the illuminated side. And the transduction is the photoreceptor changing shapes and starting a cascade of events, including the release of auxin in that one particular side. And keep in mind that the perception happens in one location, but signaling pathway carries that information to the area that then responds to the stimuli. So, and in this particular tropism, phototropism, actually not all the steps in the signaling pathway are known yet. So this is one thing I love about biology, is that there's so much information that we don't know it all. And so this is just one example of that. So the response is that the cell wall loosens and then remodels on the shaded side of the plant. And this is something I've already shown you, but this, just as a reminder, this is going to be where this is on the side away from the light is where we'll have the concentration of auxin being the highest. And because of that, our auxin stimulates our proton ATPase. Protons are pumped into the cell wall. Expansins are activated, which are enzymes that basically chop up the cellulose cross walls, and then the cell can elongate. And so the result of a bunch of that happening all on the one side is that our plant then grows in the direction of the light. Okay, let's talk about gravitropism. This is the response of a plant to the direction of gravity. So gravitropism in stems is a negative response because the stems grow away from gravity or up. And it is a positive tropism in roots because the roots grow down or towards gravity. So how does a plant know which way is down? Well, in specific cells, there are amyloplasts. We've mentioned this before in the roots, for instance, when we were talking about that in the root cap area. And amyloplasts are colorless plastids that contain large amounts of starch, and so they are heavy. So they fall in the direction of gravity. And so it's similar to crystals in your ears that allow you to know which way is down. But other things tell you that you're upside down too. Like if somebody flips you upside down, those crystals in your ears will fall in the direction of gravity, letting you know which way is down. But you also can feel it by your vision, by the pressure in your head, that sort of thing. So we know that plants use amyloplasts, but certainly they use other things as well. So you've seen this particular image before, and I'm going to focus on root gravitropism when we're discussing gravitropism. So I wanted to bring this back up again so that it looks familiar to you. So remember, these are the columella cells, parts of the root cap, and there are usually 3 to 4 tiers of them, and that's what contains the statoliths or amyloplasts, however you want to say that. Those are sort of interchangeable in most discussions, and so that's what falls in the direction of gravity. So that's where the perception of gravity occurs. So here we go. So starting on the left, if we are looking at this, here's our regular plant, and just looking at these columella cells in the root cap, and all the amyloplasts are down here at the bottom. And then we rotate it 90 degrees, and now all these amyloplasts take about a minute or two to fall down. By 2 to 3 minutes, they've settled out, and now the plant knows this side is down, and it will begin to allow the roots to grow down. So the amyloplasts the amyloplast settling tends to be part of the perception of gravity. So immediately after gravity-stimulation is what we call it, which means a change in direction, if we look at the pH of the cytoplasm within these rows of columella cells, there is a change. So in tier 2, let me grab my pen. Right here in tier 2, once we rotate the root, immediately there is a gradient of pH, so a difference in the cytoplasmic pH in this cell versus the one down here. And the one at the bottom is more acidic, meaning it has more hydrogen ions, than the ones towards the top. So gravity sensing occurs at the root tip in these cells, but the growth response occurs in a different area of the root. And this change in pH is thought to be one of the early signaling events, or the early part of the transduction pathway that leads to the change in growth further up the root. This is just the very beginning stages. There are all kinds of different ways that signaling takes place. So these pH changes, there's been shown to be electrical signal changes throughout the root as well. And so all of that is part of the transduction pathway for root gravitropism. So if you remember these 3 zones of the root, we have our cell division, elongation, and maturation zone. When we're looking at this, the root cap is down here, so this is where gravity perception takes place. That information has to travel all the way up here to what we call the distal elongation zone, so the top of the elongation zone, and that's where differential growth occurs for gravitropism. So, like I said, the signal transduction pathway contains a lot of things. We definitely have the early signaling pathway of the cytosolic pH change across across tier 2 of the columella cells. There are tension on the cytoskeleton, ionic signal is generated, and we again have lateral transport of auxin. But auxin has the opposite effect in roots than it does in stems, and it actually inhibits cell elongation in roots. So when we look at this particular root, so normally we have a strong amount of auxin coming down and then equal flow in the both directions. Okay, so there's no gradient across the root, but once we turn it on its side, as auxin is coming down, more of it goes on the bottom side than the top side. So there is a gradient of auxins being set up. Now, on the higher concentration side, which would be the side closest to down, there is an inhibition of cell elongation. and the top portion continues to grow at its regular rate, and the resultant growth is down. So if we look at our change in elongation rates of the cells at the response site, like we said, those on the bottom grow less, those on the top grow more, and so this is our response portion of gravitropism. So roots grow down in response to gravity, and of course that allows them to acquire water and minerals much more efficiently. So what happens if we place plants in space? Because in space we don't have gravity. So the image here on the left is grown here on Earth, and the one on the right is grown in the space station. And you can tell that the one on the right doesn't exactly have a particular direction that these roots are growing in. Here's just another example of one grown in space, so you can take a look at these crazy roots. So what we've learned is that without the presence of gravity, roots grow towards humidity. So if there's an area of higher humidity concentration, that's where the roots grow to. But if the humidity is even throughout, the roots tend to grow in a spiral pattern. So why is that important? Why do we care? What's the big deal about plants in space? Well, the reason for that, why we're interested in it, is that if we're going to do long exploratory missions to Mars, which NASA is intending to do, it's going to require food, clean air, and good mental health of our astronauts. And plants have been known to do all of those. So fresh foods as opposed to freeze-dried are much healthier. And just the act of gardening or tending to plants has been shown to increase the mood and mental health of people. We also know that plants tend to clean the air as they are absorbing the carbon dioxide. It makes the air nice and clean. So we need to understand how plants respond to gravity stress, basically. Because in order for a crew to go all the way to Mars, it's going to take about 2 to 3 years, and that's a very long time. So plants are going to be absolutely necessary. So as far as plants in space, we are really just in the beginning stages of understanding space agriculture. When we first started really cultivating plants here on Earth, we've had a long time to have experience with that. Now we know what we're doing, right? Remember, we still were learning things during the Dust Bowl about how to make sure that we were planting correctly. So in space, it has different challenges. Gases and liquids don't act the same in space as they do on Earth. So how can we make plants successful in those environments? So that's why more and more research is needed. So I would very much like for you to go and take a look at these two videos. Just so you know, the one on the bottom will include a plant called Arabidopsis. That is like the model plant for plant genetics. The whole entire genetics has been sequenced, and just so I can feel old, that was completed at NC State when I was a grad student. But that's the plant that most people use to do experiments on because we know all about its genetics. And so when we start manipulating its environment, we can see how it responds. If you see Mr. Simon Gilroy, who I do love, I trained with him. He taught me a lot of techniques for my research, and in case you haven't figured it out by now, my research was the one that involved the early signaling pathway to root gravitropism, looking at the changes in cytosolic pH. So you'll love Simon. He's very funny, old hippie, just a really great guy. So do go watch these videos. You'll love it. And then to end with, if you haven't seen The Martian yet, you need to. I love that movie because the botanist is the hero. How often do you get that? So anyway, Hope y'all enjoyed today, and I will see you next time.